Okay, so hello everyone. Welcome to this new edition of our talks for accessible genomics. Our speaker today is Dr. Lara Urban. Dr. Urban is a research fellow from the Gemmell lab at the University of Otago. She has a recently published paper, which she will be talking about in this talk. So I let her introduce herself more in her talk, and so I'll hand over to you the floor, Dr. Urban.
Thank you very much, Khalil. I just see that quite some people are still entering, so if you could continue just admitting them, that would be great. Yeah, so I will take it slow in the introduction so that everyone can slowly join. In general, I would love to have this like as a very relaxed meeting. So please feel free if you have a question in between to just unmute yourself and interrupt me, and we can talk about it. Maybe if it's a bit more general question, we can discuss it at the end, but you can choose. Cool. Let me share my slides. Thank you. So can you see that, Khalil? Just thumbs up, perfect, great. Cool, so hi everyone. As Khalil just said, my name is Lara. I'm currently a Humboldt Research Fellow at the University of Otago in New Zealand. And I'm at the moment focusing on conservation genomics. That means I'm mainly using statistical genomics and quantitative genomics to understand how we can use genomics to support the conservation of endangered species. So I'm mainly focusing on two critically endangered species here endemic to New Zealand, the takahē and the kākāpo. But ideally, the sort of framework that I'm developing is applicable to like any endangered species in the world. I'm also at the moment involved in some environmental DNA research where I'm still using the M9 and nanopore sequencing a little bit. So if you're interested in any of these projects, you can either write me on Slack or contact me afterwards in any way. So please feel free to do that. But so the project I'm going to talk about now was actually conceived during my PhD back at the University of Cambridge and the European Bioinformatics Institute in the UK. And the project is quite special because it was a group of PhD students who got together and planned this project together, like all next to their normal PhD project. So my PhD project was in statistical cancer genomics. The other people had like different projects, partially focusing on the Tasmanian devil and so on. So really different projects, but we all thought, we want to come together and use M9 sequencing for something. Because at the time that we started coming together, like a lot of people, a lot of researchers still thought that, the M9 has such a high error rate, nanopore sequencing in general, that it can't be used for any real research yet. And we were sort of interested in to which extent this is true and what we can actually find out with nanopore sequencing. And it's very good today that I know all of you who are here are really interested in nanopore sequencing, so I don't have to introduce the M9 or nanopore sequencing itself. But what I want to talk about specifically is about how we can use nanopore sequencing for freshwater monitoring. So I'm presenting the project by the organization that we called PuntSeq. So these PhD students I talked about right now, we founded PuntSeq. It's now 3 years ago, I think, to find out how we can use nanopore sequencing for freshwater monitoring. And as I said, it was like really just a project led by PhD students, and we acquired funding from the University of Cambridge, and we acquired a PBSRC grant, but in generally we had quite little funding, like all just sort of startup grants that we used. Why we wanted to use the M9 was because of its portability. So our idea was like with the portability, you could easily go to any remote area in the world and do freshwater monitoring there. So as you know, the M9 like has quite some advantages, like you can also sequence in real time, you can get epigenetic profiles with it, you can get the super long reads. But I think the portability that it was so small, like made us be very interested in it. So as Khalil said, our study was just published in eLife. And I think Khalil is going to share the link to the paper. So if you want to know any more details, please just read up on that or just contact me and ask. So, but why did we call the project PuntSeq? So some of you might not actually know where this name is coming from. So the name comes from punting and sequencing. Punting is the sort of sports or activity that you can see here. So it's like a very specific way of traveling on a boat, and it's a really popular way of seeing the city Cambridge when you go there as a tourist. So you can like hire one of these punts, and then you get one of these sticks. Oh, sorry, you cannot actually see my arrow right now. So you get one of these sticks, and then you just push yourself forward with one of these sticks. And the punting is just something very characteristic for Cambridge. So we thought it would be funny to have this idea, hey, we take the Minian onto the boat and do the sequencing directly on such a punt. But obviously it was just like a sort of an idea that we had to also make citizens interested in the project, to do like a lot of outreach projects and so on. In general, we were interested, how can freshwater monitoring really be brought to more remote areas in the world? Considering that the access to clean fresh water, to clean drinking water, has even been named one of the United Nations Sustainability Goals for 2030. So it's like really a worldwide problem to try and understand, is fresh water safe to drink, safe to swim on, and so on. This illustration here, by the way, like is part of a really nice outreach article that the EBI, including Spencer and Oana, wrote about us. So you can Google for that if you're interested in a more outreachy article. So having said that, like that we want to bring like freshwater monitoring to remote places, obviously there is also a problem in the UK itself with freshwater quality. So if you just look at this, the Times article from actually 2019, so not a long time ago, the Environment Agency of the UK found out that no river in England is safe for swimming. And actually more than 80% of the river didn't reach the minimum standards of the EU. I guess at that time the UK was still a member of the EU. So like even in the UK, a really developed country, there are a lot of issues with freshwater quality. So as said, we conducted our study in Cambridge in the UK. Sorry, I'm having a brain here. Yeah, so I'm just showing this map here. It's like I know that a lot of people from the Southern Hemisphere are joining. You can see, now my arrow doesn't want to work now. So you see Cambridge is here, like in the UK, very close to London. I also always, whenever I use a map now, I try to include New Zealand on the map because, as you might know, New Zealand is often being excluded from the world map. So whenever I show a map, I try to include it here. And this is actually, can see where I myself live at the moment. Yeah, I don't know, my arrow keeps disappearing down here. So Cambridge, as you probably know, is very well known for rowing on the River Cam. So here you see like one of the accidents that happened during one of the years that I was rowing myself. I mean, obviously it shouldn't look like that, but just to show you, like the River Cam is actually a really tiny, very narrow river. So not perfect for rowing, but still, because the Cambridge people had to compete against Oxford, I guess they started rowing on this river. But also people who live in Cambridge use the River Cam in very many different ways. There are like people who are swimming in it. There are people who are fishing in it. There's a lot of other boats and rowing boats. There's even like this competition at the end of every university year where the students build like their own boats made out of cardboards, and they try to survive as long as possible in their cardboard boats as a competition, so to sort of celebrate that they finished the university year. So you see the people in Cambridge get in touch a lot with the water from their river. So the question was, what is the relationship between the citizens of Cambridge and the river? So the very first thing we did within the PuntSeq project was to conduct a public survey amongst nearly 300 participants. And we ask, like, at what location do you actually get in touch with the river? And as you see here, so we here see the city of Cambridge. A lot of people are actually getting in touch with the river upstream, which is around the Grantchester Meadows area. And this is where most people are going swimming or fishing. And then we have like 40% who are like being in touch with Cambridge, like directly in the city where they are doing the punting that I just showed you. And then we have like people even further downstream where there's rowing activity and so on. So this is like sort of a little bit the picture of the city of Cambridge and how humans interact with it. And then we asked people, how often did you like, get any diseases from the river? And actually what the response was that 1 out of 5 participants of the survey said that they had obtained an infection likely attributed to the contact with the water of the river Kem. So this will probably be very biased because obviously people who think they have had a disease from the river Kem will take part in the survey more probably. So it wasn't like a properly like standardized study, but at the same time we realized, wow, there is really a need for investigating what's swimming in this river, because nothing was really known about which pathogens could cause these diseases. And the symptoms that the people would get were like fever-like symptoms, wound infections, and this so-called swimmer's itch, where you get like red dots all over your skin. So this definitely showed us, okay, even in the UK, there is a need for freshwater monitoring. And they said, like, no one, not the environmental agencies, not the local hospital, no one could tell us which pathogens cause these sorts of infections. So that's why we said we are going to take an environmental metagenomics monitoring approach. As you probably all know, metagenomics just means that you can sequence all the DNA that you find in a sample. If you apply it to environmental samples like soil or water or even air, you basically just sequence all the DNA that you find in these samples and can like that determine which species occur there. So in this study, we were particularly interested in bacteria. I know that many of you are interested in using nanopore sequences for sequencing for viral detection, and obviously that can be done as well. But we in this case really thought, because it's known that so many waterborne diseases come from bacteria, such as cholera and leptospirosis, that we thought we are for now going to focus on bacteria. I still hope that for those of you who are only interested in viruses, some of the protocol optimizations that I'm going to show you now will be helpful. So what did we do? We went out to the river at 3 different time points. So we went once in April, once in June, and once in August, and started from like downstream of the river. So that is the location 9 here that you see, and took like water samples, always 1 liter of water at all these different locations. So that means we have like a really nice and spatio-temporal picture of the river Kem in this 1 year. In addition, we always included a negative control, which just means ultra pure water, into our sequencing to see that we don't have like any sort of contamination in our data. What we did then was we would like filter 400 milliliter of river water of these samples through a cellulose membrane. We would then do typical DNA extraction using a standard QIAGEN kit. We would then do 16S RNA amplification, so specific bacterial 16S RNA primers, and would also, like we use the barcoded primers, which means that we can actually barcode up to 12 samples on the very same flow cell. Because as I mentioned at the beginning, we didn't have too much funding for this project, as it was a purely student-led project. So we tried to reduce the cost as much as possible. And then we used the like standard Oxford Nanopore Technologies ligation kit that we applied to the pooled barcoded reads to then get our libraries, which we would then sequence on the nanopore. And I'm sure that most of you are used to these processes. So we had like these 9 different samples. We also took a replicate from sample 9 just to see how comparable the results would be if you more or less at the same time take a water sample at the same location, but you separately run them through all of these processing steps. So that means we had 10 samples, and then we included the negative control, and then we also included a positive control based on a mock community, but I'm going to talk about that in a second. So we basically had 12 samples per time point that we could then throw together onto one flow cell. So, and now I just want to show you where we went from there. So after the sequencing that I was just talking about, by the way, if you're interested in that, we just ran the sequencing as long as we could. So it was up to 48 hours, because sometimes even at that time point, there were still a few reads coming through. And obviously you don't lose anything if you afterwards apply stringent filtering approaches that would remove any low quality reads. Then we applied base calling to the reads. We used what at that time was the definitely best base caller out there, Gapi, with the flip-flop option that is even improving the read calling quality at like locations where you have a lot of similar consecutive bases. And I do think Gapi is still amongst the most widely used base callers right now. It definitely was at that time. We then used PortJob to do the demultiplexing, meaning we would assign each read to its respective barcode and would like that know where this read was coming from in our sample. And we also use PortJob, to remove all additional adapters that were still attached to our reads, so that we in the end had like our pure 16S rRNA reads. Then we would need to do taxonomic classification to actually understand which bacteria occur in there. And here I quickly want to show you in detail what we have done. So as you see here, I write here that we used Minimap2 with a k-mer size of 15, and we used the SILBA database, which is a specific 16S RNA database. But as you probably all know, there are many different tools out there, classification tools, and there are also different databases that you can use. So when we started like investigating the options that are out there that you can apply at that stage, it was quite crazy. So we thought, oh, if like other researchers will run into the same problem, it would be good to give them an idea of how these different classification tools compare. So that was our first like really in-depth task, to benchmark these different classification tools. So as I told you before, our 12th sample on the flow cell was a positive control. So there was a mock community that we ordered from Simo Biomics. So it's just really a bacterial community where the different species and the different compositions are known. So in our case, we had like 7 different species in this small community. So we would actually know when we sequence it and apply different classification tools, how well do these perform and actually reflect what we know is the truth. So I would like really, whenever you plan a sequencing project, advise to include both a negative and a positive control if it's the first time that you are applying a sort of protocol. So what you can see here is that, as said, we know the bacterial DNA content in our positive control. We then, as we did with all the other samples, apply the 16S RNA amplification and the sequencing on it, and then use different classification tools that we could find out there. And because, as you see here on the right column, we know what the compositions of these different species should be, we can calculate, and we are here using just a typical root mean squared error, we can calculate what the deviance from the expected composition is and which classification tool performs best. So to show you an example of one classification tool, so I'm showing the Minimap2 tool here again. What we get at the top here is we can plot the observed abundance versus the expected abundance of the bacterial species. And I'm already doing this here on the bacterial family level because all of the other analysis that I'm going to show you now, he also performed on the bacterial family level first, because to go like further down on the bacterial genus and species level, it always requires some additional like confirmation steps, which I will also show you later. But for now, we are staying on the family level. However, it was like straightforward here because each of the 7 species I just told you about belonged to a 7 family. So the more community is based on the fact that all the species are very remotely related. So what you see is that we have like a huge over amplification of Enterobacteriaceae, or in this case, it was E. coli. So this could be because of the choice of the primers. So the 16 sRNA primers that we used have been modeled according to E. coli sequences. So it might be that the primers are just more efficient for this species. And we thought a lot about how we could deal with this problem to actually, compare our classification tools. But the really best solution that we ended up with was to just exclude Enterobacteriaceae from this analysis for now. And this is the plot that you see below to see how the relative composition of the remaining bacteria that have not been amplified by the primer perform. And here for Minimap, you actually see that the performance is quite good in terms of comparison of observed abundance and expected abundance. And based on this, we can now calculate the root mean squared error. And I'm just showing you a table here that compares the root mean squared error. So if you just look at that last 2 lines, that's the root mean squared error, including Enterobacteriaceae, and then the reduced one that is excluding Enterobacteriaceae. And we have like many different classification tools that you see in the different columns at the top that we compare with each other. And obviously, the smaller the RMSE is, the better the classification tool performs. So we see here that ID Taxa and Minimap were like amongst the best performing tools that we found. Obviously, there is also a huge difference in the actual runtime and memory requirement of these tools. So we also did a comparison there. And ID Taxa, whereas it performs really well, it had a crazy like usage footprint on the memory and runtime. So we just concluded that ID Taxa would probably not be very useful for like, remote locations where you might just have a laptop and would want to do all your computations on there. So this is why for us, Minimap2 was the best candidate after these analysis. We, however, also validated this not only on the on this positive control, but also on a freshwater sample. So what you see here is that we benchmarked the very same tools on a freshwater sample. Obviously, in a freshwater sample, we don't know what the expected bacteria or their compositions are. So what we decided to do is to do a majority vote. We would like per read, like compare across all of these classification tools, what the majority of the classification tools would classify this read as, and would then compare again against across these classification tools, which of them would actually predict the majority vote candidate. And you see again here that Minimap2 is actually performing very well, again with together with ID Taxa, also together with Bingo and MapSeq. So especially MapSeq, I have to emphasize as well, performed quite well in both cases and also doesn't have like a very large memory and runtime requirement. So that can definitely be an alternative. So, but I want to actually jump to some results now. So now we have our Minimap2 classified sequence reads. And these are now the number of reads that we were able to classify onto the bacterial family level. So we have the April flow cell to the left, the June flow cell in the middle, and the August flow cell to the right. And then you again have like the different locations and the negative and the more community. Firstly, luckily, we didn't find nearly any reads in the negative controls, meaning that we didn't have any contamination, like neither in the lab, like due to our handling, nor due to any like sort of kits that we would use, because obviously like these DNA extraction kits and library prep kits, they actually use bacterial content. So controlling for contamination is, especially for bacterial analysis, very, very important. So you see that there's a high variability between the number of sequence reads that we get per flow cell. In August, we get more than 5 million. In June, we don't even get 1 million. And we are still not sure why this has happened. It can partially be explained by the DNA concentrations that we put in. So we can definitely recommend try to get as high DNA concentrations as possible, but it can't be fully explained by that. So we also think that the flow cell type, so that was the 9.4.1 that we used at that time, might have still been a bit more variable and unstable than the nowadays flow cells of the version 10 that are supposedly a lot more robust than back then. You also see that a few of the samples like get really, really few reads, which is like a big problem, because if we only have a few reads per sample, we can't actually compare the sample with another sample that has a lot more reads, because the chance of detecting especially rare bacteria is a lot higher in the sample with more reads, so that we can't directly compare them. So that was like actually our biggest problem, this difference of read numbers. So this is where we are now coming to the next step in our bioinformatics consensus workflow. So we were just down there at the taxonomic classification, and now we are coming to the rarefaction, because to deal with this problem of different numbers of reads per samples, that is what we chose to do, downsampling or rarefaction, whatever you want to call it. Basically saying that each sample, we will only randomly choose a number of reads and the same number of reads per sample to make the outcome per sample comparable. At the same time, obviously, if we take away like a lot of reads from the very same sample, we might actually change results. We might change the diversity index and the general conclusions. So what we did is we ran like a lot of rarefactions, drawing randomly a certain number of reads. And this is what you see here on the x-axis over there, the sampling depth, and would then see how many bacterial families do we still get. And you can see that obviously, the more reads you maintain, the more families you get. But you also see that there's a plateau building up, like nearly a bit like a logarithmic function, where we see that from roughly this place on, and we chose that this would be at 37,000 reads, you actually don't gain so much more from having more reads. Meaning that if we downsample to 37,000 reads in a sample, we nearly maintain all the families, all the family diversity that we had seen originally. I also confirmed that by running a mental test on the rarefied and the full data set. And we also see here on the right that like the richness within these samples is like mostly maintained. So you see here the percentage of the original richness. So we are losing like a little bit in a few samples, but yeah, like in general, more than 80% of the richness is maintained. And now you might ask, why don't you just set it higher, the threshold of the reads, so that we can actually maintain more reads? The problem is all the samples that have less than this number of reads, we have to throw out. So we basically want to find like a threshold where we can maintain as many reads as possible, like, and across the samples, because by throwing reads, whole samples out, we are obviously also going to lose reads. You already see that actually from our overall 30 samples. So these were the 10 different sampling points that we had times the 3 time points. we only maintained 16 in that case. So there was definitely like, a weak point of our study, that we would have liked to maintain more samples, but we could have not compared all of them with each other if we had maintained them. So let's now look into some results of the actual bacterial diversity. So here at the top, you just see the different numbers of reads that we received per sample. And here on the x-axis, like always, the different samples as you would see them along the River Cam stretch. And you can also see here, you have the legend here, how many could be classified in general, how many of them could be classified at the bacterial family level, and so on. But let's not focus down on that right now. So that's an image, that's a figure from the paper if you want to look at that in detail. But what is more interesting is that, so here down here, I'm only showing now the samples that had like more than 100 reads in general. And then the ones that had like less than the 37,000 reads that I just said we used as a threshold, I'm just showing them in a transparent way here, because we are not yet really going to use them. But what's the important message here? We are plotting the 10 most abundant bacteria here, bacterial families here. And you see that like, so all the others that are not the 10 most abundant families are here, the green ones on the top. But you see that like nearly the entire composition of all these samples is made up of these 10 most abundant families, showing us that these are some sort of core microbiome of the River Cam that that take over the majority in each of the samples. To look at this a bit differently, we took the relative bacterial compositions across the samples and the different families and plotted them as a heat map, which you see here. So we have like the different samples up here and the different families of bacteria here. What you see is that we have sort of 4 different blocks. So we call them the clusters C to 4, which you see color coded over here. And this one here, this is the cluster 3 actually. You see it only occurs in the 3 samples over here, that if we go up here, you see that these are the dark gray ones, which are the mock community samples, which makes sense. So these are just the bacteria that occurred in the mock community, just a proof of principle. It's showing us the mock community is obviously different than the samples. But what is really interesting is that we have like these 2 core clusters down here. These are the clusters 2 and 4 that again represent our core families, the core microbiome that occur like across all the different sites that we sequenced. And then we have like a lot of other bacteria that sort of differ between the samples. Also interesting to see our replicates from number 9. So these are the 2 replicates cluster really nicely together. So showing that the replication actually worked. And even in such a, like, you have to think, we are talking about a really small local scale here, like a river stretch of like 10 kilometers, yeah. But even within that, we can see highly localized signals within this already localized study, which is quite cool to see. So here I'm just plotting these bacterial families from these 2 cores, which we now call the 2 clusters, sorry, which we now call the core microbiome of the River Cam. And you see that the 3 most abundant ones are Burkholderia-ceae, Spirosomaceae, and this NS1112 marine group. All bacterial families that include like genera like Limnohabitans, Rhodophyrax, Aquabacterium, that are all very well-known freshwater bacteria, which shows us that we can like that we at least the main bacteria that we find make a lot of sense that they occur in freshwater because they have already been known to do so. So we can also look at the distribution of the data differently by plotting a principal component analysis, which I was doing here. So we just again took like the relative reads of the bacteria across the different locations and time points and plotted them as a PCA. You see here that I'm plotting the 3rd against the 4th PC because I'm just telling you the PC1 against PC4, and we are showing that in the paper, but it doesn't result in anything very interesting because it's dominated by one outlier, which is the sample 7, but I will talk about the sample 7 later on. So, but what is interesting is that these consecutive principal components show us another structure. And that structure is on the PC3 here, a slightly temporal one, with like the April samples clustering over here in the positive range of the principal component, and the June and the August sample are clustering over here. So we have basically the spring month and the 2 summer months over here. So what we can do is you can see into which bacterial families contribute most to this variation that is captured by the principal components. So if we look into the contribution to the principal component 3, we see that like we have the top bacterial families, Carnobacteriaceae, Saprospiraceae, and these are bacterial families that are known to occur more often in colder, relatively colder environments. So obviously it's like you can't make any sort of conclusion about that they might have occurred only in April because April was colder than June and August, because also a lot of other factors differ, like chemical measurements, pH and so on would differ as well. So it might not actually be the temperature that has an effect on that, but we can definitely see that we see like some temporal differences in our samples. So, but like after showing you now like sort of the core microbiome and the general patterns in the data, I would like to talk about pathogens, what might interest you most. So what we did first, so we now moved to the bacterial genus level, because obviously you can think working like on the bacterial family level is quite hard for identifying pathogens, because every bacterial family might contain some pathogenic species, but then also species that are not pathogenic. So you never really know what you get. With the genus, we are already a little bit more accurate. You might think why didn't they go down to the species level? So we were quite wary with like the still quite elevated error rate of nanopore sequencing to go onto the species level just per default without actually looking into our reads. So this overview analysis, we still did it on the genus level. By the way, we estimated in our flow cells, but as I said, these were the old versions, that we had an error rate of around 8% in our reads. So what we did is we chose a genera that are known to be a potential pathogens, or to include a lot of pathogens in pathogenic species in their genus. And you see that we have like here the different genera. Here we have like the number of reads that we got across all the locations. And here we have the sort of pathogen that these bacteria are classified as. So we have like waterborne bacterial diseases. So these bacteria here are associated with that. We here have drinking water associated pathogens. We here have potential drinking water pathogens. So these are a little bit, now my arrow is again not moving, so yeah. These are like, yeah, not as ascertained as the drinking water pathogens. We have human pathogens. And then what we were also interested in was wastewater associated bacteria, because what I didn't tell you beforehand, at one location at the river Cam, we have like a wastewater outflow coming into the river, and we were interested if we would see any sort of signal from that. So you see like all the wastewater associated bacterial genera, we have them down here. So now we again have the different locations and the different time points here across our river. And I know that this figure contains quite a bit, so please feel free to look it up again in the paper afterwards if that goes a bit too fast now. But what we see here is, so the different circle sizes, they represent the relative read at this specific location. So how much reads of this location are actually assigned to this specific bacterial genus. Whereas the color of the circles represents the standard deviation to the like mean relative read number of this genus across all of the reads. Yeah, so if you see that this is like a really deep red color, it means that like it's unexpectedly a lot of this genus occurs at this specific location. So we see that quite a lot of red dots are coming up here, like at the end, which means downstream of the city Cambridge. Especially here at the sample 7, which I had told you about beforehand, it was like a proper outlier, maybe because so many potentially pathogenic bacterial genera are showing up that don't occur at all anywhere upstream. And then we also have like the sample 8, where we see that a lot of the wastewater genera start showing up. So this was definitely interesting for us, because obviously one would expect that potential pathogens might develop more downstream of a city because of all the like wastewater effluent that is coming in. I mean, there's in Cambridge, there are like a lot of houseboats that don't have like proper sanitary outflow. There are a lot of cows that are like standing on the meadows. So there's really a lot of inflow from the surroundings that could cause this spike in potential pathogens. And yeah, as I said, especially sample 8 here, like with all the wastewater genera, was really interesting as well. And before I go into detail with this one now, I just wanted to show you that we also did like a lot of hydrochemical studies on the very same samples, especially one of our colleagues, Yotis, who is a real expert in that. And this figure here, I'm just showing you to say that even the hydrochemical profile confirms that our sort of a pollution of the river Cam increases downstream as opposed to upstream. So here you again have the coloring. These are the upstream samples, the downstream samples. And we have now superimposed them on this existing data on the 60 largest rivers in the world. And what you have to know here is that so what is plotted here are basically like natrium corrected molar concentrations that are representative of how polluted a river is. Because if you see that the dark, the filled in circles, let's say, they are like potentially clean rivers. Whereas here, these circles here are the like anthropogenically impacted and polluted rivers. And by clustering them like that, you can see how polluted a river is if you superimpose your own data. And indeed, you can see that, so here for the river Cam, we have like just as shown as ellipses here, the April sample, the June, and the August here. So June and August are again highly overlapping. And we are just, for an example, showing the August sample here, that we see that within the August sample, like the downstream samples here are lying a lot closer to the actual polluted rivers than the upstream samples that lie closer to the like non-polluted rivers. So really something serious seems to happen while the river is flowing through Cambridge. And I just tell you now that the same signal with this downstream upstream we also find in the April month and the June month, but we couldn't plot all of them on the same plot because you wouldn't see anything anymore after all. So that just to show that, yeah, we definitely think we found something meaningful here. But let's go back to our actual sequencing data. So you see that we find like quite a few interesting genera here. Arcobacter, by the way, is a very well-known freshwater bacteria that leads to gastrointestinal infections in humans, which have often been reported in connection with the river Cam. But we also find other genera where we also know they also contain species that are not pathogenic at all. So maybe it's just the non-pathogenic ones that we find here. So here we really understood we have to look down into the species level. So this we now did on a per genus level. So what we did is, and I'm showing here an example of Legionella, we would get all our sequences that were assigned to Legionella and would pool it together with database sequences from Legionella. both pathogenic Legionella species and strains, as well as so-called saprophytic Legionella strains. So these are the ones that are not pathogenic at all and are expected to occur in like a normal environment. So we then did a multiple sequence alignment with them, and as you can see here, build a like phylogenetic tree and visualize them like that. So I will go through it slowly so that you understand what's happening here. So this is the phylogenetic tree of all the sequences, yeah, our sequences and the database sequences. Now the outer ring here it shows you the database sequences. So you can see that here, the reference in the outer ring, with the violet ones being the pathogenic ones, and other, so non-pathogenic strains being the green ones. Plus we also included like one outgroup that you can see here. You can see that for Legionella, we have like one a pathogenic or like it's not only one, but a few pathogenic strains here that we got from the databases. The rest of them are actually like non-pathogenic ones. And now the second inner circle, so these are just our reads now. These are the different time points that the reads were taken from. And then the inner location are the different locations to see if there's any pattern there. In short, what we see here is that like our reads cluster like together with a wide range of different non-pathogenic Legionella species. Nothing is clustering together with this with these pathogenic reads that lie in here. What is probably suggesting that all the Legionella reads that we find are just saprophytic and not pathogenic. And like this will probably not lead to any danger while swimming in the river. I will show you another example though from Pseudomonas, or Pseudomonas, I think you say in English. So again, see the outer ring. We have like the green, the different green strains. And we have like, again here, a pathogenic strain or species. So it can be on this species or strain level. And again, we are clustering our reads here in the inner circle. And you see that here, this part of the tree that is like indicated by this violet background here, fall very closely together with this pathogenic strain or species in that case, with this Pseudomonas aeruginosus, showing or suggesting that these reads might actually point towards the presence of this pathogenic species. And if you look into the inner ring, you again see that most of the samples that contain this specific species are downstream samples, again pointing towards the increased pollution of the downstream samples here. So we thought that this idea was quite cool. Instead of like just doing across the board species assignments, which might be incorrect due to the still quite high error rate of nanopore sequencing, looking down in detail into our reads and see what they suggest, even on a like sort of biological, sort of evolutionary level. So now if we go back again to the same plot, I want to point you towards one thing that when I saw it, I was like really struck by it. We find Leptospira genus in the water. Leptospira is the bacteria that can cause the leptospirosis that I was talking about beforehand. That can be a really, really severe disease. So there was this very famous case, I don't know, like I hadn't known about it, but apparently it was quite famous, where Andy Holmes, who was like a gold medal rower, English gold medal rower, he died from leptospirosis in the middle of London after he got infected while rowing. So rowers are particularly exposed to leptospirosis, which is caused by water that has been contaminated by the urine of infected animals, often mice and rats and so on. If you come into contact with this water and it can get into your bloodstream, you can get really easily infected. And rowers, as I know from their own experience, always have a lot of injuries on the hands, because it goes like so difficult on the hands. So it's very easy that just some water during training drops into your hands. So understandably, the rowing community in Cambridge was always very concerned about that there might be Leptospira in the river water. And every year, even in England, a few people die from leptospirosis, which is also called Weil's disease, if you know that name better. So in the end, it affects the liver or the kidney. And if you don't recognize it early enough, several years, every year, several people even die from it. So when I saw this, and as an active rower, I knew, oh, wow, this is big. We have to follow up on it. I said, yeah, we definitely have to look into what's happening here. So firstly, I looked at the spatial distribution of the Leptospira reeds that we find. And you see here that the peak is where at the sample 8 here just the April and June examples, and you still see the signal a little bit downstream though at the samples 9. So at the sample 8, this is exactly at the location where we have the sewage pipe outlet. And if I just can show you this picture again before, sample 8, we find all the wastewater associated bacteria, which makes a lot of sense because they've been used in the wastewater sludge and they are now being flushed out. But as you see on this picture, we also see like a huge relative increase of the Leptospira genus reeds that obviously come, and you see this here on the picture, out of this sewage water outflow. Very interestingly, at roughly the same time, a lab in Berlin had observed the very same thing. So they used 16S RNA sequencing, but using PacBio to compare the bacterial composition upstream and downstream of wastewater plants. And they found that both Legionella and Leptospira were enriched at the outflow rather than de-riched, what should be the case, at the outflow of the wastewater plant, which was really interesting. And obviously, we here find something very similar, that the outflow of a wastewater treatment plant, we find this huge signal of Leptospira, as we don't find it anywhere else in the river, except for its direct downstream location. So we repeated the very same analysis that I just showed you with Leptospira. And with Leptospira, we have the nice advantage that we really have quite a lot more pathogenic strains and also quite a few here saprophytic strains. And these are just classified as other environmental strains by NCBI. So definitely also non-pathogenic. And here I find you see the clustering idea a little bit nicer with our samples. So you do see that we have the pathogenic clade out here, but it doesn't really cluster if you look here with any of our reeds. It's really sort of its own clade. Whereas the remaining reeds, like the saprophytic reeds, cluster more clearly with our reeds here together, pointing towards that the reeds that we have found from Leptospira are probably only saprophytic Leptospira reeds. And hopefully, we also still think, I mean, we haven't proven it finally, but we suggest it's not pathogenic. But as this is obviously public health concern, we said we have to go further with this. And we sent the very same samples that we sequenced to Public Health England, who have a standard, which they call leptospirosis enhanced surveillance protocol, that they usually apply to blood samples to see if people are infected by Leptospira. But they changed the protocol slightly for us to apply it to our water samples. And they could indeed confirm that the Leptospira that we found was only saprophytic and not pathogenic, which was a really nice sort of confirmation of our approaches. So to sum up, I hope I haven't talked too long. I haven't timed this before it because I thought maybe people ask questions in between anyways. But in summary, so we here report a quite simple low-cost workflow to understand freshwater quality by sequencing different bacterial communities. With low cost, I mean that we estimated that if you would by now use one of these 96 sample barcoding kits that Oxford Nanopore Technologies provide now, you could achieve sampling and like all the costs at £20 per sample. We only, as said, used the 12 sample barcoding kit. And as you saw, we already like that, we had to throw some out. Some samples out because we didn't have enough sequences. However, as said, with the improving flow cells, I'm sure this problem of like, missing reads per sample will be resolved as well. Plus, there are really promising techniques out there at the moment that allow for balancing sequencing. You might have heard of this selective adaptive sequencing approaches. So these ones might allow us to really say, hey, I'm having one flow cell and I want to allocate exactly the same sequencing effort to each barcode that I could give in, what would be very promising. So this is really a proof of context study here that shows that nanopore sequencing of 16S RNA is actually exact enough to understand like the general bacterial composition and patterns in any sort of freshwater system, and which also allows to detect pathogens. So we really envisioned to have like this full framework that combines, I said, like cost effectiveness, which it had to be for us because we didn't have a lot of funding, with like a really fast sample processing and fast data processing. And what was also important for us was the data transparency and also the transparency of the analysis. So our data is all available, publicly available. All our code is publicly available on GitHub. So you can easily, just take the data and like recapitulate everything we have done. And hopefully you will find the same thing at the end. So, and I think our benchmarking especially profited from like the evaluation of the different computational classification tools that I showed you. But we in general advise like on the complete workflow that you have to take from water sampling to the end bacterial classification and interpretation. And yeah, as said, we hope that our study will be useful when it comes to environmental sampling in any sorts of settings, also remote settings. And as we have learned during the last Accessible Genomics talk, there are definitely like approaches on the way that try to make everything, not only the sequencing approach, feasible in remote locations. This was actually our plan at the beginning as well, to like really develop every step remotely, but then we just didn't have enough money to like buy all these like portable devices. But it really seems as if this was becoming possible at the moment. We were also really keen on involving citizens because ultimately we really think tools like the Minayan can help democratizing science, in that more and more people can start doing science themselves and actually understand the outcome of it afterwards, instead of believing any fake news that occur on the internet. So what is next for PuntSeq? So PuntSeq still exists in Cambridge, and I'm still in close contact with the team. They are actually just building it as a permanent university society, so that every student who comes to Cambridge can get their hands onto the Minayan and can get to do some sequencing. So we also intend to promote UK and also global citizen science projects more in the future. So we've set up a website where we advertise all sorts of events that we have planned. We also made a small introductory video about nanopore sequencing that you can watch on there. We are going to a lot of outreach events in pubs, in universities, in schools to talk about our project. We organized a whole weekend workshop where we actually taught citizens like everything from water sampling, as you see here, to the wet lab, to like the data analysis, which we ended up doing in a pub, how it's properly done in UK, I guess. So it was like a really, really nice experience to see how within a weekend, you can teach people everything they need to know to conduct these analyses themselves. Yeah, and we are also like doing lots of interviews and other sort of outreach articles to keep people informed. So methodologically, we hope that we can benchmark novel types of nanopore tools and protocols that come out, including for sure things like the fungal that promise a lot cheaper sequencing techniques, and also trying whole genome sequencing, something you might have wondered about along the way, why we haven't tried shotgun sequencing yet. So this is also definitely something that we've already started. So we have collected shotgun sequencing data from the very same spots that I just showed you, and are now doing one-to-one comparisons, how 16S and shotgun compares with each other, also price-wise though. And yeah, and ideally we would like to get involved in freshwater analysis around the world, and are obviously very interested in sort of giving the knowledge and the protocols and analysis and everything we have done now to others who are interested in doing the same, and who might not have to start off at exactly the same starting point again where we have started. So with that, I would like to thank all these people, and even more people who didn't fit onto the slide anymore. But I have to mention Maximilian Stammnitz and Andre Holzer in particular. So these are the guys you see here on the picture over there, because it was basically the three of us who really drove this project next to our PhDs. And it was a really fantastic experience, doing everything from, project planning and financial acquisitions to publication in the end, what only happened like a short while ago now. And also sort of becoming friends with your science colleagues on the side was really good. And here are all our sort of funders and supporters listed as well. So I hope I didn't forget anything too important, but I'm happy to chat now as long as you guys want. So thank you very much for listening.
Thank you so much, Dr. Urban. That was very fascinating. And I have to ask, your data visualizations were beautiful. What software did you use?
So actually, like the basic plots themselves have mainly been made in R. And then, just fitting them together afterwards in Inkscape and so on. For the trees, the phylogenetic trees, we used ITOL, which is a really cool tool where you can nicely visualize your multiple sequence alignments. But I also have to, again, emphasize Andre's contributions here, because he is really, really good with the data visualization. So I think that was fantastic. Yeah.
Cool. We have some questions in the chat. If you could, one is maybe Dr. Lara, if you could ask directly then.
Yeah, sorry. I'm attending 2 meetings now.
Sorry, I could read it for you. Yeah.
Yeah. Okay. Thanks.
Yeah. So the first question is, what can you say about grab versus composite sampling?
So this is obviously relating to wastewater sampling, right? So the only thing we did was to sample downstream of a wastewater outflow. So grab and composite sampling is being done when the wastewater actually comes into the wastewater how do you call it? Wastewater plant, right? Where you would like either just take like quickly one sample out, or you would actually do composite ones across time. So so we didn't do that, right? We didn't do wastewater sequencing. So I'm actually involved here in New Zealand now into the national wastewater monitoring program, because as you guys might know, in New Zealand we don't really have COVID right now. So I'm saying not really, because just yesterday 2 community cases were detected that came out from a quarantine facility. So something hasn't worked very well there, and they still don't know what happened. But in general, all across the country, we don't have any coronavirus, which means that for us, the sewage sampling is like really a great tool, because as others have shown, you can detect COVID in wastewater before you can detect it in the community. So in a place like ours, where we don't have it in the community, it's amazing to use it as a parallel tool. Having said that, I'm more involved on the computational side of things for this project. I, however, know that we are using composite samples, but I wouldn't know about how they compare. So, yeah.
The next question here is if you have plans to explore Biomics as well.
So not specifically just Viromics. So we are, as said, doing shotgun sequencing at the moment, with which we should discover viruses as well, obviously. But I mean, that will be interesting to see how much can you really discover with shotgun sequencing with respect to viruses and fungi and eukaryotes, and how is it more helpful than 16S RNA, also with respect to finding functional genes and so on. But it's not that we only want to look into viruses. I mean, I think also we should not forget that hundreds of thousands of people are dying every year from waterborne bacterial infections, right? Like, I know that like I know, at least like I have always lived in countries where that was not a huge problem. But obviously in other countries, this is still a really, really big problem. So I don't think we should like just go into looking for viruses now, right? Bacteria are also still very, very important.
Do we have
Hi,
other questions?
Lara. Good morning. I have a question. For instance, if we are to perform the same 16S approach or other similar metabarcoding approach using certain specific markers, do we need to use like specific PCR kits, like in terms of Taq polymerases, or can ordinary Taq polymerases be used for the enrichment part, or do we need to, use more efficient polymerases for that?
So I would assume that the polymerases and everything, you can use the very same one. So you can just substitute the primers. The one thing I'm not sure about right now is, so we use the standard 16S RNA kit provided by Oxford Nanopore Technologies. And they are providing the primers that already have like the motor protein attached, which allows the read to actually go through the nanopores. So if you wanted to design any other sort of primer, you would basically have to do it, I guess, through Oxford Nanopore Technologies. I'm at the moment not aware which other primer sets they are offering, but I would assume that they are going to extend it a lot, because I've recently seen many more papers about sequencing 12S, COI, and so on the nanopore. So this possibility seems to be out there. There's just something to be cautious about. If you just used your own primers, they wouldn't have the motor protein on it yet. So you would have to like look into how you can design that, if that makes sense.
Yeah. Thanks, Lara.
Sure.
Hi, Lara. I have a question. By the way, I'm Paul. I'm the resident bioinformatician of Philippine Genome Center, Mindanao. Um, your study actually reminded me verily of my own master's thesis. So I did metagenomic sequencing of seaweed farm sediments. So I used Illumina-based platform a few years ago. So when I saw your like heat maps and graphs, I was really reminded of my own thesis as well. So my question is, did you have like I don't know if I missed it in your talk, but did you like find any correlations between like for example um temporally like for example in the months of April and June there might have been more outflow of wastes? We like consider that in your
Yeah, so we tried to take into account as much as we could. So we measured like, biochemical and physical parameters, including pH, temperatures, different ions, different anions, and so on, to understand if anything like that might have an impact. But it was, given that we only had 3 data points, quite difficult to disentangle the impact of these different parameters, because they are obviously like correlated with each other a lot as well, right? So we couldn't say why to achieve, like in the UK, obviously. So we hope that with that we could at least minimize the impact that we would get, from like outflow from the fields outside. Sometimes you have cows in the fields, sometimes not, and so on. And we thought if we at least sample at a tri-period, if that makes sense. Does it answer your question? Or
yeah, it's like if like there are more sewage activities during, for example, these certain months compared to these months. But yeah, you explained that with your few data points, it's difficult to disentangle all the factors. Yeah, I
and I
understand.
mean specifically with the sewage outflow point, as I showed, like across our samples, so across the time points, we consistently see that like specific bacterial genera come up across all the time points, and that like downstream of the sewage, we also still find like some leftover of the signal, basically telling us, it's not something that we have just missed somewhere, but it's really there, and we can even detect it downstream still.
I see. Makes sense. Yeah. Thank you.
Thank you.
I have a last question. Sorry.
Sure.
Since these are communities, we expect that there are like interactions. For instance, keystone species that could also inhibit proliferation of other bacterial communities. How do we analyze that in metagenomics data? Because I read a paper about pathogenic leptospires that states that they speculate that we couldn't really expect to detect pathogenic leptospires in certain types of water because certain conditions prevent it from proliferating, or other bacterial species also prevents it from proliferating. So I'm interested if we can do that analysis using metagenomics data.
Yeah, so we will definitely find all sorts of interactions, obviously, with the metagenomic data, because we will find the relative abundance of all the bacteria. I guess the question is, what are you interested in that? Are you interested in like understanding a functional interaction? That might be difficult because you might have to control it in the lab, right? Because as I just said, you have so many different factors and also so many different bacterial taxa that will slightly change the composition, that it might be hard to do functional interactions. It might be better, superimpose the assumptions that you want to make. Having said that, I think by just seeing the output, so sort of the end point of the interactions, that is already enough for what we want to understand at any time, or PuntSeq in general. And as said, like if PuntSeq can help accessible genomics in any sorts of ways with any pipeline optimizations and so on, just be in touch. We would be very happy to have some international collaborations that go beyond the river cam.
So if there are no other questions,
I think we'll really take you up on that, especially on the data visualization. That's really very cool.
Thank you very much. Yeah.
Great. So I hope we'll have further conversations after this, maybe in Slack or over email, and hoping that there will be collaborations after this. So with that, I'd like to thank Dr. Urban for this great talk, and I'll be uploading this in YouTube. So and see you guys in Slack and wherever we're discussing this. Have a great day, guys.
Great. Thank you, everyone.
Thank you. Thank you.